people on the internet say they have debunked you, how do I know what's true? You'd have to be more specific. They've debunked what? They've debunked that you can hook amino acids together, the two amino acids, that one has a side chain carboxylic acid, one has a side chain amine, and they've put that together. That's never been debunked. Has that person gone to a blackboard and shown you the way that could happen? If not, then that's not been solved. Has a person gone to a blackboard and shown you how two glucose molecules could couple together with a discrete coupling, but only that coupling that's needed? Nobody's ever debunked that. Has anybody ever shown the polymerization of oligonucleotides without getting the 2,5 linkage and without getting the branching? Has anybody ever done that? If not, I've not been debunked. Has anybody ever been able to come up with, where did the code come from? Where did the code come from? Has anybody ever done that? If they won't go to the blackboard and show us that, it's not been debunked. Has anybody ever said that they could take the discrete pieces from a cell and put a cell together? Has everybody ever said that? Has anybody ever done that? If not, I've not been debunked. So when you get down to the exact things that I've said, I don't think I've been debunked. So that's how you know. Isn't it true that the origin of life field has made significant breakthroughs, such as in the RNA world hypothesis? In my opinion, the RNA world hypothesis is a hypothesis on life support. RNA is terribly unstable. And RNA, you want to make RNA that's going to code for peptides, and you want to make the ribozymes. The ribozymes, they can act as enzymes, but they're not as good enzymes as proteins. And they're highly unstable. They are unstable in water. You have to have the prescribed code. How you get all the pieces and how you take all the different pieces to hook up to be in a prescribed order to do anything. And as Steve Benner, an RNA expert, will point out, that if you just make random polymers of nucleotides to make RNA, that you will have far more, like thousands of times or hundreds of thousands of times more RNA molecules that degrade other RNA rather than synthesize RNA, so they don't copy themselves. You have much more of them degrading RNA. And RNA is just terribly unstable. So let's say one RNA molecule, even though the chances are way more difficult than 1 in 10 to the 50th. 1 in 10 to the 40th means that you wouldn't have enough time in our universe to do this. So it's a very small probability that you'd get one of the right sequence to do anything useful. And even the ones that quote unquote duplicate themselves, look at the work that's been done on this. So there's so much priming, so much that's gone. And I've interviewed Rob Statler on my YouTube channel. And you'll hear about this, where we went through one of these experiments by Gerald Joyce of all that he had to do to get duplication. And usually you never get replication more than about 10% of the RNA. And it's a very painstaking process. It's nothing that would happen on an early Earth. And you're not dealing with one molecule. You're taking billions upon billions of molecules of that same RNA to try to do something. When you're dealing with one molecule, you only have, at the most, under the best conditions, hours before that thing would degrade. And so you had to have polymerized this in the absence of any other molecules. You couldn't have amino acids around because they would stop the polymerization. But you do have to have an amino acid hooked onto one of the ends. So that's a mystery on how that happens. And so you get these RNAs, and then it has to be totally pure in order to get this condensation polymerization. Now, all of a sudden, you want it to be exposed to a bunch of amino acids. This thing's going to cleave. It's in water. It is going to leave. It's not going to last long. You have hours, which is the twinkling of an eye when you're talking about prebiotic chemistry. So the RNA problem is a huge problem. And it's very difficult to work with. So the RNA hypothesis, I would encourage you to read this new book that's out, X-Ray of Life. I mean, he just stacks up. He goes through 100 pages or 200 pages just on the RNA world hypothesis problem. So that's not a very good hypothesis anymore. According to the paper, Creation of a Bacterial Cell Controlled by a Chemically Synthesized Genome, haven't scientists built a cell capable of replication? No way Nowhere close. You can take an existing cell and modify it. In other words, you can, people have put in a modified genome, modified the genome, or taken a genome from one cell and put it into another. But the whole cell was there to begin with. To try to put a cell together, nobody's ever put a cell together, nowhere close. So think about this hypothetical experiment. Rather than trying to make all the building blocks, why don't we just take a cell, which we can do, and we can deconstruct a cell. So you can take the lipids, you can take the the polypeptides, not even going down to the amino acids, just take the polypeptides. So you already have all the enzymes. Take the polypeptides, take the RNA, take the DNA, all of these structures, and put them in discrete bonds. You want to take everything, all the different pieces, and now just say put it together again. I've already given you the code because I've given you the DNA. And I've already given you the polysaccharides, so you already have the code in that. I've already given you the polypeptides, so you have all of those enzymes. Can you just construct a cell? Nobody knows how to do this because there's so much regulatory information that is needed in a cell, so many non-covalent interactions that need to be properly aligned. Nobody knows how to do that. So to think that people have constructed a cell, this shows how confused the general public really is. It's a real problem. If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together and I will share with you about why I embrace the resurrection of Jesus. Is abiogenesis comparable to speculative theories like time travel, faster than light travel, or teleportation? I wouldn't put it in the same category as those. I would put it that, yeah, there's a lot of speculation going on, but on things that like time travel, teleportation, it's hard. These are difficult problems to even think about how you would address this. I think origin of life is really difficult on how you would do this, but at least you can go in a lab and test this. You can test your ideas, and you can see that your ideas really don't work. That you can certainly do. Doesn't opposing origin of life research seem unscientific considering the potential for future discoveries? Would you like to keep going down rat holes? Isn't it good to turn people away and say, that could not have been how life was made? Or should we just say, keep doing it? Life could not have happened that way, but just keep doing it. Just keep messing around. No, it is good to correct them. And that's what I'm doing. I'm correcting them. I have repeatedly said, I think one day we will figure out how to make life in the lab. That's one question. And that's a much easier question than how did life start in the beginning? When you have to start with small molecules and build the whole thing up. That's a whole different thing. So I would say a much more relevant thing is, can we create life in our laboratory? That's a much easier thing to be able to do. And I think that to point people out the problems that they're having is a good thing. That's what we do. That's the way science is. We don't just let people keep going down the wrong way without pointing it out to them. Why do you primarily discuss abiogenesis in churches or religious settings? Actually, I don't. I primarily talk about the Bible when I'm in religious settings. I am asked sometimes to address this because people are thoroughly confused about this. And what happens is people will somehow use this to say, hey, we understand how life was made. There's nothing really magical about this, and therefore somehow try to negate God. Well, first of all, if we did know how life formed, it wouldn't negate God at all. It makes God all the more magnanimous when we understand something about how he did it. So in other words, prior to the 1950s, we didn't understand how genetic information was transferred. And then we learned from Watson and Crick that the code is in the DNA. That didn't make God any less. That makes God even more in my eyes, because I'm like, oh, so that's how you did it. So in other words, to point these things out would not ruin it, but people try to negate God. So I'm invited to give talks. And when I give those talks, I film those and I put those up on YouTube. But generally, every week I'm in a church speaking about the Bible, and I'm putting those up on YouTube now. So I don't primarily do that. I primarily don't do that, speaking about origin of life things. in churches. Isn't specified complexity a term associated with intelligent design as described on Wikipedia? Well, Wikipedia is wrong. The term specified complexity was used by Leslie Orgel and others in the 1970s, talking about how Leslie Orgel, an origin of life researcher who's very well known in the field, he's passed away now, talked about the types of information that you would need, segregating that from Shannon information is the information that you can pull out of random signals. But specified information predated even Leslie Orgel. That is a term that was used by information theorists when they were talking about Shannon-type information versus specified information, which you need a lot more coding for specified information than Shannon information. So that term was never used by intelligent design people until much later. So if Wikipedia had said that to you, Wikipedia is wrong. But Wikipedia has occasionally been known to be wrong. So you can go and correct that now. You've criticized origin of life researchers for failing to create lifelike systems. Given the complexity of the problem, what specific experimental milestones would convince you that meaningful progress has been made? To convince me that meaningful progress has been made in the area of origin life. Again, origin of life is separate from can we create life? Can we create life is a much easier problem to address, in my opinion, than origin of life. Because origin of life, you have to start with an early Earth condition. You have to start with these early Earth conditions, and you have to have life somehow spring forth rapidly, soon after the Earth cooled. Immediately after the Earth cools from the heavy bombardment, you have to have life. It springs forth. That's a very difficult question. Nobody was here. Making life in the lab, we're here. And we can push things. As scientists, we can push things into place. We can take things from living cells and use it as part of our building block for other cells. We can do this sort of thing. So it's a much easier problem. I think that just making life in a lab is ultimately going to come first, long before we figure out how all this happened on an early Earth. And so what are meaningful experiments to me to make life? Well, people have taken, can you take a living cell? This is what would mean a lot to me. Can you take a living cell that has died, so it's just died, and then bring it back to life? That would be a meaningful experiment to me. That, again, has nothing to do with abiogenesis, how life originated. But can you even just take a dead entity and bring it back to life? That would tell me a lot about our ability to possibly someday make life, make a living system from non-life to life. So that would be a great experiment. Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org. Thank you so much. Are there any origin of life theories or experiments you find scientifically promising, even if incomplete? How would you suggest improving current research methods? Again, origin of life is a very tough problem. Just go ahead and make life. Go ahead and make life. And it's much easier to do a resurrection than it is to have an ab initio synthesis. Take something that has just died, so you have everything pretty much in place. Try to bring that to life. I think that would be an amazing experiment. That would really be amazing. That'll win a Nobel Prize quite quickly, I think. And then you can begin to understand, what is it that life has? What is it that life has that is this key to life? It's a very interesting question. Haven't researchers demonstrated polymerization under simulated prebiotic conditions such as wet-dry cycles and mineral catalyzed reactions? Yeah, I told you the wet-dry cycles, you get racemization, And how big are the wet-dry cycles, the polypeptides that they make? And you don't just get the amine coupling to the carboxylic acid at the alpha position, the amine, and just you get the side chain. Here's the problem. So you have an amino acid and it has to hook to another amino acid, but about half of the amino acids have an extra group hanging off, and an extra active group hanging off, and that extra active group ends up participating in the polymerization. So you get what is not an amino acid anymore. You get something that's not an amino acid. So nobody has ever solved that problem. And you will see most people that'll go through wet-dry cycles, they don't choose any one of the 10 amino acids that can cause side chain reaction. They conveniently only go with a selected 10 and not the other 10. That's not what life does. So they'll select it out, and then they never check the racemization problem. And now if you look at what's been done on mineral surfaces, yes, this can catalyze reactions to very low molecular weights. And then you get the side branch. Same thing happens with RNA. You get the side branching problem. This is, for example, what Steve Benner showed. You get these side branching problems. Again, and you go in with very pure material. What happens is these guys want very pure material when they want to do the polymerization. But they don't want pure material. They want to have amino acids present if they want to say that RNA then started making polypeptides. How? How do you exclude everything else and get a pure compound, but now when you have to react it with something else, just that compound comes in? You're not using the gross mixtures that would have been available. Every organic chemist, every synthetic organic chemist sees exactly what I see. It's not just me seeing these things. It's every organic chemist who sees these things. And they concur. This is a huge problem. This is a huge problem. Why won't the origin of life people engage with me? Why will they only send a YouTuber to engage with me? They won't engage with me themselves, not even virtually. They won't even engage with me. Why? Why is it when I do have a discussion, like I had with Lee Cronin, he wouldn't answer any of the questions that are put forward to him? Why? Why is that? These were very relevant questions because every one of those has to be solved in order to go toward life. These are the most basic and fundamental questions. And so these polymerization reactions that have occurred, these have occurred under very strict conditions because to make a polymer, to make a condensation polymer, you have to have very pure material. I mean 99.99% pure. How do you get that? You don't get that from the reactions that you've done in your own lab. You don't get that from, and if you say, well, it happened in a crystallization, okay. But now that has to mix with other amino acids that themselves happen by a crystallization and others that happen by a crystallization. It's hard to get crystallization when you have vast mixtures of compounds around. They obscure things. So the chemistry becomes very difficult. You've mentioned issuing research challenges to scientists like Lee Cronin. Have you considered collaborating with these researchers directly to combine expertise and address scientific problems in prebiotic chemistry? I've been asked by them, not by Lee Cronin, but by others, to join on with them, but I don't see that they're going in any right direction. I don't think what Lee Cronin is doing has anything to do with origin of life. I don't think his assembly theory tells us anything about life's origin. What it does is it tries to quantify how unusual a compound could be, and he says, does it have a causal history? Some other people would mean the same thing and say, was it intelligently designed? But he doesn't want to use that word. He wants to use the word causal history. You could take causal history and exchange it for intelligent design and it'd mean the same thing. But his causal history, I can look at a molecule. As a synthetic organic chemist, I can look at a molecule and tell you whether it had a causal history if there's more than one of them. You can have one molecule form in blasts that are really quite unusual molecules. But if you have multiples of them, if you have 2 or 3 or 4 of them, then that starts becoming a very difficult question. That's exactly what Lee's assembly theory is saying. And he gets this by blowing molecules apart in a mass spec, which is a very, very silly way to do. Mass spec was never made as a tool for this sort of thing, to blow molecules apart and say that's how I'm going to define complexity. It's much better when you do as an organic chemist can do, and as his team can certainly do. And you look at the number of stereocenters, you look at the complexity of how things are arranged, and if you have multiples of them, then the complexity index goes way up. I can look at the same thing and tell you, you have a group of those. Those had, in Lee's terms, a causal history. In Discovery Institute's term, those have an intelligent designer behind them, because it's too complex to figure how you're going to get multiples of those made. That assembly theory wants to put a number on, fine. But tell me about how two amino acids come together without side chain interaction. Tells me nothing, absolutely nothing. And Lee confessed that. Lee confessed that at the Harvard debate. This has nothing to do with polypeptides coupling. Even though he used polypeptide coupling in his paper, he says he wasn't talking about that. So maybe he's got Velcro balls sticking together, but it has nothing to do with chemistry. How you get polysaccharides hooked together, that's a huge problem, huge problem. How you get RNA hooked together with the 3' 5' coupling rather than the 2' 5' coupling, or without branching. Again, how you do that, his assembly theory tells us nothing. Have I thought about working with him? No, I haven't thought about working with him. First of all, he wouldn't have me work with him. But second of all, I don't know how to go about solving these things. But I can tell you that the way many people are going, it is screaming out, that is not giving you the answer. All right, given the complexity of origin of life research, how do you think scientists should communicate uncertainties and progress to the public without oversimplifying or overhyping? That's a fair question. I mean, how do you communicate to the public? You communicate to the public, I think the truth of it, we set it up under these conditions to think of this going on an early Earth. I can't think of an example where you would find this on an early Earth. It'd be very hard to have this, because on an early Earth you have vast mixtures. Say it like it is, but when this thing gets overhyped, that's why I show these studies that talk about how 2/3 of the public thinks that scientists have made life in the lab. That's how much it's been hyped up. That's the problem with this whole thing. So the overhype is a real problem. And then the expectations, where people have said, we will have life in 2 years in our lab, we'll have life in 3 to 5 years, we'll have life in 5 years. I mean, these are key people. Lee Cronin has made that claim that he'd have life in his lab in 2 years. That was in 2011, he said that. Jack Szostak said he'd have life in his lab in 3 to 5 years, and that was in 2014. Dimitar Seselov says they'd have life in the lab in 5 years. That was in 2014. He said that. All of them miss out on these dates. And then Jack Szostak, to his credit, a few years later said, we haven't even made the RNA. I mean, from RNA to life is a long, long way. I mean, it's multiple regulatory systems you have to hook up here and have them all working in concert that we don't even know how to define in a simple cell. We haven't even defined this stuff because there's so much we haven't even learned yet. And they've made these broad claims, and Lee, to his credit, has said that projection of 2 years was overambitious. So it's just been overhyped, and we want to stop that hype from going forth. You want to listen to hype all day? No, it confuses us. What experimental conditions do you consider most plausible for prebiotic chemistry? Are there any specific studies you think come close to demonstrating a possible path to life? People have talked about these hydrothermal vents. These hydrothermal vents, it's a bunch of nonsense, I think. Because if you start to get structures, these would hydrolyze, these would break up any polymer you have. These things are quite hot. You get racemization. This is all in water. I mean, water is tough to do the polymerizations. And you want the prebiotic, you want the hydrothermal vent to give you the energy to do these sorts of things. ATP, the way a cell gets energy, it's under these very strict conditions. It's very hard to get life under these sorts of conditions. What experimental conditions are most plausible? Most plausible is you do nothing, in the sense that you want to put a bunch of chemicals together and just watch them. I mean, this is what early Earth was, but it wasn't sugars and it wasn't piles of enantiomerically pure amino acids and piles of enantiomerically pure carbohydrates. It wasn't that, and piles of nucleobases. It wasn't that. It was very small molecules, like maybe methane, oxygen, CO, CO2, N2, maybe some ammonia. Although a lot of times these don't last with high UV around. And so you start with that and see what you can do with that. It's very difficult. But if you want to start with these others, you can't put them under these very strict conditions. You'd have to start much milder. So I think that most people would say that for origin of life, again, it's a hard problem. I would go back and say, can we make life? And then the easier question is, can we start with something that was alive, a cell, and bring it back to life? That would be the main experiment I think that we probably should be working on now. And in our lab, not trying to reproduce early Earth conditions, because again, that problem is too difficult. Right now, maybe in 500 years it won't be. Do you believe the scientific community has set unrealistic timelines for origin of life research, or is the issue more about how progress is communicated to the public? Well, they communicate their progress to the public saying we're going to have life in 2 years, or we're going to have life in 3 to 5 years, or we're going to have life in 5 years. Yeah, that's a problem on how they communicated to the public. It's a problem when you can read every month, probably every 2 weeks, you can see another article, key milestone in origin of life studies. And I look at it and I'm like, this is nothing. This is nothing. This does nothing. So yeah, I think it's a real problem in the way it's communicated. And I don't think it's being done very well. And I think the source of this is actually the origin of life researchers themselves, and then it's ramped up an order of magnitude by the press, and the researchers don't pull it back if they get the chance to. What would you say is the single most critical missing piece in our understanding of life's origin? Polymerization, metabolic networks, or something else entirely? Polymerization is a big problem. Even if you want to say meteorites brought these compounds here, the things wouldn't be, you can't do condensation polymerization reactions on mixtures of compounds. It just doesn't work. And so you have to go in with 99.99+% pure compounds in order to do polymerization. Yeah, polymerization is a big problem. And your wet-dry cycles, your heat cycles, don't do it because water hydrolyzes these things. These are condensation polymerization. Water sends them back, and water prevents them from going forward. The free energy is positive here. Metabolic networks, wow, that's a hard one. Metabolic networks, you get a little thing spewing out of a vent in the ocean. People say, hey, this is the beginnings of metabolism. That is utterly ridiculous. Metabolism has so many discrete functions that need to go on. That's another hard problem. So missing critical pieces, like everything is missing. What are the critical pieces missing? Everything is missing. I mean, organic compounds sitting together form what Steve Benner calls tar, asphalt. They form junk. And every organic chemist knows this. Every one of the reactions they've seen at times, every day, they see things go to junk, tar, polymeric goop that's totally unusable. Organic reactions have a tendency to do this. These are big problems that need to be solved that have never been solved. Some argue that abiogenesis research should be viewed through a systems chemistry approach rather than as individual steps. Do you believe this holistic approach has merit? Yeah, I mean, origin of life, you're going to have this amazing choreographed dance going on. You can't just take these subunits and just pack them all together. You can't do that. You can't just pack these all together and expect them to work. It's not going to happen. You've got to be able to have discrete chemistry going on. And there are many systems that have to work together. Nobody knows how to do this. So these are big problems. Yeah, you're going to have to have this a systems chemistry approach. But we have no idea how to do this. We don't have, and let alone on an early Earth where there was no brain working on them. Hasn't Lee Cronin and Sarah Walker developed a theory that explains the origin of life? The answer is no, not even close. Nor do they say that it explains the origin of life. Their theory talks about a causal history. They will look at complexity and they'll say it has a causal history. Their theory has never explained the origin of life. Never. It can't. It doesn't do that. Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.